thank you for asking me to talk again. This means either they didn't understand anything that I said during my first talk, or they want to hear the update. So to make both people happy, I am going to review what I covered last time for the first. The first slide shows the title Green Disinfectant Surfactant Foams to mitigate as well as kill virus and microbe spreading. And I want to again emphasize, can, can you see my arrow too here?
No, but that's okay. But
Okay, okay.
Jesse
So,
will need to advance the slides, I guess.
Yeah,
okay,
Jesse will have to do everything.
okay. So right at the bottom, I am again saying that the best way, my aim is to limit or mitigate the spreading. That's the best way to control the virus. And one way, of course, to use the Indian namaste at the bottom, or bowing, that the Asian one, rather than shaking hands or cuffing into the elbows.
Disinfectant foam.
Hello? Yeah. So next slide shows that the disinfectant products used currently for COVID-19 can increase the risk of chronic lung diseases because the bleach is used. Bleach fumes can hurt lungs, and with ammonia, like in Windex, it can also irritate, uh, produce chloramine gases which are irritating. And there are other problems. They irritate skin, throat, nose, etc. So the idea is not there. We have two ideas. One is to use the foam which will stick to the surface and kill the virus, rather than just throw some kind of liquid which will drip. The other idea is to substitute for bleach the green surfactants. So next slide. So it shows that the current methods that are used, for example, on the top left, you see the hazmat. If you drip some solutions onto that, it will just drip. If I show a demonstration, you don't see me, right? You can't see me?
I can see you at the top of your screen.
Oh, okay. Others can see me, because then I could demonstrate what I mean. Otherwise, you can just imagine.
I can see you some too on your video.
So let me just try whether it works or not. So if I just put water or something here, it will just drip. It will just drip. It wouldn't stay on the surface. See, I don't know whether you saw that. If you didn't, you have to just take my word for it. But if I use a foam, like in my shaving cream, it will stick there. So it will stick there. And you can control the sticking time by controlling the surfactant composition and all that. That's the whole key. So you can have the foam stick and kill the virus or microbes. You can have it sticking there for long enough time. I don't know if you saw the demonstration anyway. So that is the whole idea. Instead of using the spray solution like you see at the bottom left. So the application of bleach solution, it drips. The other problem is that when you use bleach solution, it produces a lot of toxic waste solution. And this is the problem during Ebola. We started this project during the Ebola time. So what do you do with all these toxic solutions? And that's a big problem. Whereas if you use foams, you almost have no liquid. And also if you have a lot of toxic, it will wash to the seep to the underground. And using also the solution water, you cannot get into crevices on the ceiling or under the carpets. Whereas using foam containing silicones, modified silicones, you can get to cracks and everything. Next slide. So this shows a simple first high school thing about how virus is being killed. Most of you, this is nothing, but let's see whether it works. Probably it won't work on Jesse's. It
I
works.
yeah, I can't. It just goes to a
Okay.
yeah.
It works
Sorry.
on my screen, but anyway. So what it will show is that how this surfactant will go and rupture the outside layer of the virus and kill it. And so that is what that demonstration would have shown. It's actually a high school demonstration, so you can imagine what it will do. Surfactant will go and absorb on the outside cover of the virus or microbe and rip the cover apart, and so exposing the virus and kill it. Okay, next slide. So this shows the dry foam, which is on the, yes, right there on the left-hand side. And the wet foam is shown on the right-hand side, which we don't want. So in the dry foam, as you can imagine, the volume occupied by the liquid material is very, very small. And also, if you introduce functionalized silicones, like the one that my first PhD student, who is a millionaire now, makes in Pune, he has a factory which makes functionalized silicone. It can penetrate into cracks, crevices, and go under the carpets and so on, which is where the virus hides. And also, by the way, when you wash your hand, if you are wearing a ring, you don't necessarily remove and kill the virus under the ring. Whereas if you have foam containing silicones, it can penetrate and kill the virus. Next slide. So this shows on the left-hand side, a conventional surfactant, sodium sulfate, which is bad for if you use on your hand all the time. I'll show the effect of that in a few minutes. And on the right-hand side, you have a foam, a dry foam. That means which has almost no liquid there. So we are trying to mix the synthetic and the green surfactants to control the foam texture so that it will stay there for the time that you want. For Ebola, it's about half an hour, and for all the viruses, probably around that time. So next slide. So this shows the principle of this controlling the stability of the foam. On the left-hand side, you all have played with soap bubbles on the beach, I hope. And the color on the soap bubbles is due to the change in thickness of the film. And that is because an arrow it takes you to the middle. It's a lamallae between two air bubbles. And if the water drains out completely from that in between the lamallae, the bubble will break. Whereas if you can control the thickness, then you can have it the foam bubble for the desired time. There is somebody called Dewar who had a film that was stable for a couple of years. And on the right-hand side, it shows the principle. If you have two layers that when they like to come towards each other, if that tail and the yellow head is surfactant that are ionic, when the two layers come towards each other, they will repel. That means it will not allow the liquid to drain out. And that's how you control the drainage time. So by using the appropriate surfactant with ionic, non-ionic, that has electrical and hydration properties, you can control the drainage time. Next slide. So this shows the use of the foam by you see the Columbia on the left hand side. This is on a shower curtain that Partha, the main worker, used his own. To get from engineering to buy any of these things is very difficult. All kinds of new regulations. So we use our own material. And you see that the foam you can also see at the bottom. So that's one other thing that you can often see the foam if it is deposited. And if it has killed the virus or not, that also is possible using the appropriate tracers. Before foaming, you see it all white. After foaming, it is now red. That means the foam is, whatever is in the foam is acting. And also you can assure there's uniform deposition by using a chlorine indicator. Next slide. So, so what we are trying to do now is use the foam sprayers. I think that's in one of the next slides too. Foam sprayers to spray the foam. There are all kinds of sprayers available, like the one that I just, you didn't see me, the shaving cream. That's that smaller scale, and foam lasers that are used to clean cars, and even reach up to the top of a aeroplane if the aeroplane is on fire. So there are all kinds of different sprayers, and we are trying to collaborate, trying to find the collaboration, people to collaborate with to design the appropriate delivery equipment. Next slide. So these are some of the delivery equipment, all the way from the smallest on the left-hand side to the largest foam laser on the right-hand side. All kinds of things are there. So next slide. So the spray benefits again: no fumes, no mist, and you can reach ceilings and cracks where the virus hides. And also you can get better adherence by controlling the composition of the surfactant. There is no splash, almost no splashback. And of course, with almost no water, there's less toxic solution seeping into the drains. And you see, of course, if you are asleep, you can look at the on the right-hand side. It's a beer that should wake you up. Look at the foam there. That's always what you want to see. And that also, by the way, is important for taste also. The texture of the foam and everything important for taste. Next slide. So what you see on this slide is that if you use if you look at the stratocorneum of the skin, in the middle that you see untreated lipids layer on the skin. But if you treat it with the kind of surfactant that you usually use, like soap, you disrupt the layer, and if you use it every day, your skin is going to be dry. And the effect of that you will also see in the next screen, one of the next slides. Next slide. So you see here very nicely what happens if we use a hard surfactant on the right hand side. Look at the leaf is all shriveled. Whereas if you use mild surfactants on the
You
left
have
hand side.
2 minutes, Som.
Oh, 2 minutes? Oh my God. Okay.
2 minutes.
Can I speak in Hindi, because then I can cover the whole thing in no time.
I'm not sure that would work.
Okay. Next slide. So that shows the effect. And this is an alien coming and saying, look, this chemical and chemical is bad for us. They cause cancer because of all the chemistry. So we are trying to use synthetic surfactant. Next slide. That's what I like to. So it's not very difficult. Use ethyl carbamate. It's toxic, but if you next slide, yeah, if you use if you remove butyl carbamate, non-toxic. Next slide. Ethyl alcohol, methyl alcohol is toxic. Ethyl alcohol, you'll all agree, is non-toxic, right? If you don't take too much of it. The point here is that you don't need much change in structure to make something benign. That's the whole point. Next slide. So our idea is here you go very fast. You use synthetic surfactants that are very, very benign, and then you mix it with biosurfactants shown in the second steps, and then you also use the dynamics, which people don't study. So we use all these principles to control the interaction of the reagents with the skin. Next slide. They're all in the nature. By the way, I follow nature. The frogs has these foams that they deposit on the eggs, which get stable for safe for days. That's the principle here. Next slide. It shows the principle of it by, it's called hinge effect. By opening the protein, hydrophilic part is exposed to water, hydrophobic part is exposed to air. It will be stable for months until the eggs hatch. Next slide. This is probably going to also show. Initially, we were used these biosurfactants to control the emulsification of the oil after oil spill. And this was covered in Stephen Hawking's Brave New World. Since you cannot see the slide, essentially what it shows that around the oil droplets, the microbes will have deposit proteins that will make the bubbles the droplets very robust, so that microbes can do their job. But that you cannot see that, but maybe once I post it, maybe you can see it. Next slide. So this also will skip because you won't be able to see it. What this would have shown is that you can pull the like the worm, silkworm, you can pull it. It's very, very strong. So now our idea has been to use the green surfactants. Next slide. Yeah, what this slide shows on the right-hand side, among the many green surfactants that we tried, sarcosinate, the glutamate, which is the third from the right, it causes minimum skin irritation. And in collaboration with June Wu at Sino Lyon, that's the one we are testing. So that's our idea is to use this surfactant alone or mixed with the conventional surfactant. Next slide. So this shows the effect of mixing. If you don't mix with zero betaine or one betaine, the skin irritation is more, but if you mix it, it is less. So the idea is to mix it so that the chemical footprint is very little and irritation will be less, but still it should do the microbial kill. Next slide. So the next one. Then the microbes produce all kinds of reactants, the surfactants. The one on the left is surfactant that we are working with, which is a biosurfactant. It has also antiviral properties. Next slide. And that shows the effect of mixing with the bleach. We want to avoid bleach. You don't want to spray bleach on your face. So this shows that you can mix the surfactant with bleach and still get pretty good surface tension properties and cleaning properties.
Som, this is Eric. We're running a bit over. Maybe
Okay.
if we could think about wrapping up.
Okay, so next slide. The last two slides shows the use of medicinal plants. They are good and they are also very disinfectant with Dev. And I think the last slide is the next one. Shows that with this material that you get from medicinal plants, you see the virus growing in the control, but on the bottom left, you see the virus is not. So there's medicinal plants also can be used. Next slide. That may be the last one. So the last slide, that was the last one. Our effect has been is to use, instead of bleach, the benign surfactants that are natural and mix it and to reduce the chemical footprint. And you also show that it kills the microbes. And viral, with Lipkin, we did some test and we'll continue to do that. Thank you.